Hi, everyone, and thanks for joining today. I'm Sarah, and I'm a senior application scientist at Roche. Today, I'll be discussing our target-enrichment RNA-seq solution for SARS-CoV-2 surveillance and genome analysis. So a disclaimer before I begin the presentation, although the results of this study are promising, the KAPA RNA HyperPrep with HyperCap workflow v3 protocol is still in development and has not been fully validated by Roche. So I'll begin the talk today with a brief introduction of SARS-CoV-2 genome research. I'll then give an overview of our KAPA SARS-CoV-2 TE workflow and panel and how we conducted performance testing of the workflow. I'll then get into what this performance was of our TE panel and workflow when testing with a SARS-CoV-2 viral copy titration. And finally, I'll discuss 3 optional workflow modifications to either shorten the time or increase performance of the workflow. So with SARS-CoV-2 research, genome research, and viral genome research in general, there are a few main research questions that people are looking at when they conduct this type of research. So in looking at a viral genome, you can determine the viral origin and evolution. This is particularly important for newly emerged viruses like SARS-CoV-2. When you're looking at viral evolution, each fixed mutated genome within a population is called a viral genome variant. Additionally, you can track host-to-host transmission of a virus. This is particularly important at the beginning of the pandemic, and a lot of the SARS-CoV-2 genome research was around that. And finally, and importantly, you can discover mutations that can cause higher virulence, pathogenicity, or immunogenicity of the virus. And these new biological properties can lead to new viral strains. So SARS-CoV-2 has an RNA genome, and this is why this requires RNA sequencing workflows to do genome and mutation analysis. Down here in the viral particle, you can see the RNA genome depicted in red in the middle of the particle. So as I just mentioned, the viral variant is defined by specific combinations of set mutations within the genome. So in looking again at the SARS-CoV-2 viral particle, if we stretch the genome out linearly in gray at the bottom, a particular protein of interest for SARS-CoV-2 is the spike protein shown in purple throughout the viral particle. And this protein is responsible for binding to the host cell receptor ACE2 for viral entry into host cells. So tracking mutations in the spike protein that might lead to better binding and then better entry into host cells is of particular importance. So if you have new viral variants with mutations in the spike gene, this can lead to those new viral variants. We see accumulated mutations in the spike gene in some of the new emerging variants of SARS-CoV-2, including the UK variant and the South African variant. So popular sample types that researchers are using to look at SARS-CoV-2 include infected host samples. And in infected host samples, you typically get a single SARS-CoV-2 genome variant present. But you can have situations where you have major and minor variants. This does occur too in long-term infected hosts, where you can have accumulated mutations that again might lead to a property that allows that variant to take over the population within the host. Another popular sample type is wastewater for community SARS-CoV-2 surveillance sequencing. And in a wastewater sample, you're more likely to have a mixture of different variants. And these can be at different ratios depending on what's dominant in that particular population. So now I'll give an overview of our KAPA SARS-CoV-2 TE panel and workflow and how we conducted performance testing. So in our workflow, we use the KAPA RNA HyperPrep kit and the HyperCap v3 workflow to enable high throughput targeted sequencing of the SARS-CoV-2 genome. So in your sample type of your viral RNA in a host background, viral RNA is shown here in green and the host background in blue. And the host background is typically a much greater proportion of the mixture than the viral RNA. So then we perform library preparation with the KAPA RNA HyperPrep kit. We do target enrichment with the KAPA SARS-CoV-2 TE panel and the HyperCap workflow v3. And then we have a final library enriched to some extent for viral sequences that's ready for Illumina sequencing. So our KAPA SARS-CoV-2 TE panel covers 184 sequences from NCBI. It covers by design 100% of the reference Wuhan genome and greater than 99.7% of another 183 genomic sequences that were publicly available. So to test the panel and workflow, we created experimental samples with human universal reference RNA and then 6 different SARS-CoV-2 genome variants mixed in. So to mimic the diversity that you might find in real-life samples, our samples contained a mixture of the viral RNA and a human RNA background. We tested multiple viral titration levels, and we had the 6 different variants mixed into each sample in equal ratios. The 6 genome variants are shown here in the table. We did have the reference Wuhan strain present, and then 5 other variants from different regions around the world. These each had SNPs and indels from the Wuhan reference, and each of the mutations were independent from each other. So our experimental overview and objectives, we set out to demonstrate performance of the SARS-CoV-2 TE panel and workflow under a variety of conditions. One of these being viral loads. So we tested from a very low viral load of 10 copies to a higher load of 1 million copies. And additionally included in our experiments were negative samples with 0 viral copies. But as these produced no valid aligned reads, as expected, I do not show any data for these samples. We also had our human RNA background, and we tested our viral titration in 2 different levels of this background, both 20 nanograms and a higher background of 100 nanograms. In testing different workflow modifications, one of these we tested was an upstream ribosomal depletion prior to library preparation. We also tested pre-capture multiplexing of 6 sample libraries per capture hybridization. And then a shortened hybridization time. So a short 1-hour hybridization compared to our standard 16-hour overnight hybridization. So now I'll talk about what the performance was of the KAPA SARS-CoV-2 TE panel with the viral copy titration. So in our viral copy titration, we had 10, 1,000, 10,000, and 1 million viral input copies in a background of 20 nanograms human RNA, shown in dark blue, or 100 nanograms human RNA, shown in yellow. We were able to identify SARS-CoV-2 reads from down as few as 10 viral input copies, and that increased from there with increasing viral input copies. Our mean SARS-CoV-2 genome coverage was between 6 and 10x for 1,000 copies, 50 to 100x for 10,000, and then over 1,000x genome coverage for 1 million viral copies for all samples. In looking at the consistency of that coverage across the SARS-CoV-2 genome coverage, our bases covered at 1x was greater than 97%, down to as few as 1,000 copies and 100 nanograms of human RNA. And then that increased to over 99% for the higher viral copy levels and 1,000 copies in 20 nanograms of human RNA. Even at our 10 viral copy samples, we were able to identify 7 to 8% of the genomic bases covered at 1x. In extending out to 10x coverage, we get again over 99% of the genome covered at 10x for 10,000 and 1 million viral copies. And then between 20 and about 50% of bases at 1,000 copies covered at 10x. And this is with 1 million next-seq Illumina read pairs. To determine how many read pairs would be needed for complete viral genome coverage at 1x for the different input viral copies, we did a downsampling analysis of the total number of sequencing reads. We're showing the results for 20 nanograms of human RNA background in the line graph on the left, and 100 nanograms of human RNA background in the line graph on the right. For 1 million viral copy samples, we achieved over 99% genome coverage in both human RNA backgrounds, so we do not show that line here on the graph. But for down to 1,000 viral copies at 250,000 reads in the lower human RNA background, we get over 99% coverage of the genome at 1x. and over 95% coverage in the higher human RNA background for 1,000 copies. Even our 10 viral copy samples, again, we achieved 3 to 8% bases covered at 1x from 50,000 to 4 million next-seq read pairs. So we then compare the results of our KAPA SARS-CoV-2 TE workflow to published data from a supplier with a similar workflow. For our lowest viral copy samples, 10 copies and 1,000 copies, we found that our KAPA SARS-CoV-2 design provides better genome coverage with the same sequencing rates spent compared to the supplier X panel. The supplier X is shown in gray, and our lower human background in dark blue, and higher 100 nanogram human RNA background in yellow. And we see 3% more bases covered at 10 copies with our workflow, and greater than 10% more bases covered at 1,000 viral input copies with our workflow. So additionally, within our lab, we tested the KAPA SARS-CoV-2 TE workflow against one of the publicly available Arctic V3 amplicon workflows that uses different supplier products. We tested both of these protocols with input samples containing 10,000 viral copies and performed analysis again on 1 million Illumina next-seq read pairs. So in the coverage plot shown here, we found generally more consistent coverage for a KAPA TE workflow across the SARS-CoV-2 genome. When we zoomed into the spike gene towards the 3' end of the genome, and we looked at the coverage across, and we normalized the coverage across the Arctic and the KAPA TE data sets, we found an area of near complete dropout to complete dropout in certain of the replicates towards the 3' end of the spike gene, while we had maintained consistent coverage in our KAPA TE workflow. So as I mentioned, within all of our viral copy titration samples, we had 6 different variants of the genome present within the single sample. And we were able to identify mutations from all 6 of those variants within a single sample using our panel. In the bar graph here, the 20 nanogram sample replicates are shown in blue and the 100 nanogram in yellow. And we're able to detect both mutations, an insertion and a deletion found within the different SARS-CoV-2 variants at near their expected frequency based on the manual mixture, which is 16.67%. And we were able to detect this in all of the replicates. So this is for a 1 million viral copy data set, so about 167,000 of each viral variant present. And while we were working on a more in-depth analysis for the lower viral copy samples for variant detection, we were able to find the most complex mutation, the 10 nucleotide deletion, was detected down in the sequencing reads to 1,000 and 10,000 copies and 100 nanograms of human RNA. And that's shown here in these IGB coverage plots by the sequencing reads that have a black line indicating a 10 nucleotide deletion in those reads. So finally, I'll discuss a few of our optional workflow modifications that we tested. So the first of these was the addition of a ribosomal RNA depletion step using KAPA riboerase prior to the KAPA RNA HyperPrep library prep. The goal of this ribo depletion step would be to reduce overall host RNA background by depleting ribosomal RNA from the host. The workflow is shown here for the ribosomal step, and then after that, you would go into the library preparation with the KAPA RNA HyperPrep kit, and then through the rest of the SARS-CoV-2 TE workflow. So when we test ribosomal depletion, we did find that it provided increased coverage for our low viral copy samples. So for this experiment, we tested 1,000 and 1 million viral copies in 100 nanograms of human RNA background. We found for SARS-CoV-2 bases covered at 1x, we get 6% more bases covered at 1x for 1,000 viral copies, and 3 times more bases covered at 10x for 1,000 viral copies from incorporating that ribosomal depletion step. Ribosomal depletion does add time and cost to the overall workflow, but it might be an option for more challenging lower viral copy samples to get more genomic data. Additionally, we tested a pre-capture multiplexing prior to enrichment with the SARS-CoV-2 probe panel to increase the throughput of the workflow. So just a consideration for pre-capture multiplex pooling for viral samples and SARS-CoV-2 samples. So we tested 8-plex pre-capture pooling versus single-plex. So prior to our target capture, 8 of our different libraries from our viral titration were pooled based on equal amounts of library. And this was done using total library mass as assessed by qubit to normalize between. So we did find, as shown here in the table on the left, that for our lower viral copy samples, those samples had less overall sequencing reads than our higher million viral copy samples, which took more of the sequencing read pool. Our recommendation around this, although we have not tested this yet, is that when possible, if doing a pre-capture multiplexing, it would be best to avoid combining libraries with vastly different sample CT values. And this would be the upstream SARS-CoV-2 viral load qPCR that's performed prior to any library preparation when choosing libraries to do pre-capture multiplexing with. And as I mentioned, the reason for this is if you have very high and lower viral load samples present in the same capture reaction, there is the possibility your high viral load samples may compete for reads. But when we did take our pre-capture multiplex samples and we normalized the read coverage down to 1 million next-seq reads across the data sets, next-seq read pairs, we found that our sequencing and coverage results were nearly identical to even a bit better with our 8-plex multi-capture versus our single-plex. And this is seen both in the mean SARS-CoV-2 genome coverage and our bases covered at 1x, the 8-plex in the gray and the single-plex for comparison in the yellow. So one last thing that we wanted to test was whether we could accomplish a faster TE workflow by shortening the hybridization time. So once the sample libraries are probed with the target enrichment panel, that reaction can be an overnight hybridization of 16 to 20 hours. So we tested shortening this hybridization to 1 hour versus our 16-hour hybridization results. So we found that a shortened 1-hour hybridization did yield similar SARS-CoV-2 genome coverage to our 16-hour hybridization at all viral copy levels. So our 1-hour hybridization results are in blue and the 16-hour hybridization results are in green. And we find there's a slight drop in mean target coverage from 10x to 8x at 1,000 viral copies, but pretty similar across the viral titration loads. When we look out at consistency of that coverage, so our 1x SARS-CoV-2 genome coverage, we find that you're still getting over 99% of the bases covered at 1x for 1,000 to 1 million copies, and from 7% to 5.8% at 10 copies. So we do find the 1-hour hybridization to be a nice balance between workflow speed and coverage, even for low viral copy samples. So in conclusion, the KAPA SARS-CoV-2 TE panel and HyperCap workflow v3 enable near complete SARS-CoV-2 genome coverage down to 1,000 viral copies and identification of even part of the SARS-CoV-2 genome from as few as 10 viral copies. We see more consistent genomic coverage than an Arctic amplicon method that we tested within our lab, and a similar target enrichment workflow from another supplier's published data. We're able to detect variants from multiple SARS-CoV-2 variant genomes within the same sample at near expected frequency. By performing pre-capture multiplexing and doing multiplex pooling in the same hybridization, you can do a higher throughput processing. Our recommendation around that is to group samples with more similar viral load prior to doing your sample pooling. And then finally, we find a shortened 1-hour hybridization offers a good balance between speed and genome coverage. Thank you for attending the webinar.